Welcome to this video on cells, structure, and function. This video will serve as a study tool for this unit. We begin with Section 1, Introduction to the Cell. The discovery of cells began with the invention of the lens. In 1665, Robert Hooke observed cork and described what he saw as little boxes, which he called cells. Anton van Leeuwenhoek in 1675 was the first person to observe living cells using a microscope. Microscopes are essential for studying cells. Magnification is the power to increase an object's apparent size. Resolution is the power to show detail clearly. This chart illustrates the resolving power line, showing the range of objects visible with the human eye, light microscopes, and electron microscopes. A light microscope uses light to pass through a specimen. The image shows the various components and how the light travels through the objective and eyepiece lenses. Here are images of Elodea, an aquatic plant, viewed under a light microscope at 40x and 400x magnification. The transmission electron microscope, or TEEM, uses an electron beam to create an image, revealing ultra-fine details inside a specimen. Examples of TEM images include the herpes virus and a plant root cell, showing internal structures in great detail. The scanning electron microscope, or SEM, provides high-resolution images of a specimen's surface structure. The SEM works by scanning a focused electron beam over the surface of a specimen to create an image. These SEM images show a mosquito head at 200x and 2000x magnification, highlighting its intricate surface texture. This image of a fly eye captured with an SEM displays the complex structure of its compound eye. More SEM images showcase different biological structures, including a neuron, the surface of the tongue, and the inside of a stomach. Other SEM examples include pollen, yeast, and various blood cells, a red blood cell, platelet, and white blood cell. This slide compares TEM and SEM by showing a virus leaving a cell. The TEM image shows the internal process, while the SEM image shows the surface. The cell theory was developed by Matthias Schleiden, who concluded all plants are made of cells, Theodor Schwann for animals, and Rudolf Virchow, who stated cells come from existing cells. The cell theory has three main points. All living things are composed of one or more cells. Cells are the basic units of structure and function, and cells come only from existing cells. Cell diversity is categorized by size, shape, and internal organization. Cells vary greatly in size. The biggest known cell is the ostrich egg. Cells also vary greatly in length and overall size. The surface area to volume ratio is crucial for cells. As cell volume increases, the surface area increases at a slower rate, which limits cell size. Cell shapes also differ widely, though most cells are roughly cuboidal or spherical. Cells are diverse in internal organization. Key terms include nucleus, organelle, eukaryotes, cells with nuclei and membrane-bound organelles, and prokaryotes, cells without them. Eukaryotes, such as animal and plant cells, and prokaryotes, like bacteria, have distinct structural differences. This image provides a general overview of a prokaryotic cell structure. This image provides a general overview of a eukaryotic cell structure. This visual comparison highlights the structural organization differences between eukaryotic and prokaryotic cells. Moving on to Section 2, Parts of the Cell. Every cell carries out essential tasks like taking food, generating energy, eliminating waste, and reproduction. Most eukaryotic cells have a cell membrane, cytoplasm, and nucleus. We will now discuss the structure and function of various organelles, including the cell membrane, nucleus, cell wall, and others listed here. The cell membrane is a selectively permeable phospholipid bilayer with proteins and cholesterol. Its function is to be the boundary between the cell and the external environment. The nucleus is a sphere containing the nucleolus. Its function is to store the cell's DNA and manage cell functions. The cell wall is a rigid wall, mainly of cellulose, in plant cells, outside the cell membrane. It provides structure and support. The cytoplasm is a gelatin-like fluid inside the cell membrane. It contains salts, minerals, and organic molecules and surrounds the organelles. The cytoskeleton is a network of thin, fibrous elements like microtubules and microfilaments. It acts as a support system for organelles and maintains cell shape. Ribosomes consist of two subunits made of protein and RNA. They are the location of protein synthesis. The endoplasmic reticulum, or ER, is a system of membranous tubules and sacs. It acts as an intercellular highway for molecule movement. There are two types, rough and smooth. The rough endoplasmic reticulum, or RER, is covered with ribosomes. It is prominent in cells that produce large amounts of proteins for export or insertion into the cell membrane. The smooth endoplasmic reticulum, or SER, is not covered with ribosomes. It is involved in the synthesis of lipids and the breakdown of toxic substances. The Golgi apparatus is a structure of stacked flat sacs. Its function is receiving, processing, packaging, and shipping proteins from the RER to other organelles or out of the cell. Mitochondria have a folded inner membrane, called cristae, within an outer membrane. Their function is converting energy stored in food into usable energy for work through cellular respiration. Lysosomes are spherical organelles with single membranes containing hydrolytic enzymes. They function to break down food particles, invading objects, or worn-out cell parts. Peroxisomes are spherical organelles with single membranes containing enzymes. Their function is to degrade hydrogen peroxide, a toxic compound produced during metabolism.